I think we'll get started with the restarting the Lecture of the Month series. And so it is my great pleasure today to introduce you to Tony Hunter and the Kinases. And I have to say, when I first saw the title, I wasn't sure if I was introducing Tony's band or Tony's lecture. And I thought, well, I should try and play it safe and go for a hybrid approach. Because I'm not sure if any of you realize this, but actually Tony's moonlighting job all the years has been actually as the Muppet Master himself, Jim Henson. And this is Tony there with Kermit the Xenopus. And basically, one of Tony's greatest creations, of course, has been Tony Hunter and the Kinases. So let me introduce you now. His CV would kill a small rainforest if you were to print it out. So I'm just going to introduce you to some of the cast of characters that he's really developed over the years and actually the profound implications they've had. So the first, of course, was Abel O. Salk. And this was really just an incredibly original finding. And what Tony discovered is actually, with a bad buffer day, a potential that no one had even really dreamed of, which is that Abel and Salk actually are tyrosine kinases, a whole new modification. And this really set the stage for growth factor signaling, oncogene pathways, and many seminal discoveries that have really laid the groundwork and are in every textbook. Of course, with Abel here, Tony's experiments in the lab and the drugs that they led to, especially Gleevec, eventually killed poor old Abel. But the world thanked him because it was actually, of course, Abel is one of the major causes of CML. And really, Tony's basic findings in the lab actually led to the discovery and development of Gleevec, which has really turned CML from a death sentence to actually a chronic and potentially curable disease. The second, of course, is CDK here. Now, he was just a tortured artist, really, when Tony found him. One day he was on, the next day he was off, and no one could figure out why. What Tony discovered, actually, it was really just the company he was keeping. It was KIP, CAP, and CYP, all of which inhibited his potential. Once Tony realized that and separated these activities and hooked them up with the very nice pair of cyclones, it was basically the key insights into the cell cycle, which again have laid the groundwork for how all of these pathways are regulated, both from yeast up to then human cells. And again, CDK inhibitors are now a very, very important therapeutic target. And again, this work has played an amazing part in that. Of course, it wouldn't be rock and roll if there wasn't some brief flings. And this here is Kit Kinase. And she's a receptor tyrosine kinase. And this was a brief but glorious affair. Damn, she nearly broke Tony's poor heart, though, and many things beside, because it turns out that he discovered that KIT, in fact, was critical, actually, for inducing the signals that maintain male fertility. And in fact, he created the first in vivo model for male sterility and the first discrete signaling pathway, actually, that gave rise to that. And finally, this, of course, is mech animal. This was really the beginning of Tony's experiments with alternative modifications. And this guy is just an animal, really. He's a kinase, he's a ubiquitin ligase, no sense of class. But really what it led to then is more recent voyages into ubiquitination, SUMOylation, and how actually kinase pathways are actually turned off. And again, these have been profound contributions. So when he's not, of course, working with the kinases, he's also a superb adventurer. He's a great rafter, and this is when they went down the Colorado River in the Grand Canyon last year. And this is mile 95, the fifth wave, and this is with his son, James. And so he's intrepid in the lab and equally intrepid, actually, in the outdoors. And this is him going over it. And finally, let me just end with saying that sometimes, when you meet, Tony's a legend, quite frankly. And sometimes when you meet your legends, your heroes, you realize that the legend is sometimes greater than the man. But in the case of Tony, it is quite assured that Tony is even greater than any legend could betray or actually papers could convey. And what I was amazed at is when I came here is that Tony has an open door policy. Right? He's like there in the lab. I mean, there he is, right? Totally accessible to me as a young assistant professor. And I can go and chat to him and talk to him. And he is, I like talking science with Tony like few other people. It's just incredible. He can not only tell you the history of the field and give you a synthesis of it, but actually point out the absolute critical point in your experiment that you need to explore. And it's just incredible and such a privilege and pleasure. I mean, I could talk to him for hours, in fact, and I don't know if it's a coincidence, actually, but it's true. But, you know, it's funny you should say that, because since I arrived, all of the chairs, except for Tony's chair, have disappeared from his office. And coincidence, maybe, right? I mean, I'm sure it's nothing to do with me. But anyway, without further ado, let me please introduce you to Tony and the Kinases.
Thanks, Clodagh. I'm not sure I can really live up to that, but anyway, it's a pleasure to start this year's series of Lecture of the Month, and Inda gave me this title, and I decided it was a fine title, so I'll, in keeping with the title, spend the first few minutes talking about my involvement with the protein kinase family, and then go on and tell you 2 stories about work that's been going on in the lab. So I'll remind you that protein phosphorylation, which is catalyzed by protein kinases, was discovered over 100 years ago now, when Phoebus Levine showed that vitellin, an egg yolk protein, contained covalently attached phosphate. And over the past 100 or so years, there's been enormous progress in understanding how phosphorylation occurs, how it's reversed, and what functions it plays. And as you've heard from Clodagh, one of the outcomes of all of this work was the development of a very successful drug for the treatment of CML, which has an unregulated BCR-able protein kinase. That was the paper, the second volume of JBC, 1906, reporting this discovery. So it was about 25 years ago now that the first sequences of some of the protein kinases were emerging following the advent of cloning and rapid DNA sequencing. And a number of these protein kinases were linked with oncogenesis. In fact, they were the products of viral oncogenes. And it was clear from this manual alignment, here BB stands for before blast, that these protein kinases all contained a number of conserved motifs. And interestingly, when these in the top panel here are all tyrosine kinases experimentally, when these were compared in sequence to the sequence of the cyclic AMP-dependent protein kinase, which had recently been sequenced the old-fashioned way, in other words, the protein was sequenced and not the DNA, it became clear that both the tyrosine and the serine-threonine kinases had these conserved motifs, which were part of the catalytic domain spanning around 300 amino acids. And Steve Hanks and I, when Steve was here as a postdoc with Bob Holly, aligned about 100 of these and found that there were these 11 conserved motifs, ranging in order from the N-terminus to the C-terminus of the catalytic domain. And gratifyingly, when Susan Taylor solved the crystal structure of the cyclic AMP-dependent protein kinase catalytic subunit a few years later, almost all of those conserved motifs turned out to line the catalytic cleft into which the ATP molecule is bound and to which the substrate peptide sequence binds. And we know from mutation of these key elements that they play a key role in catalysis. So by aligning newly emerging sequences, it became clear by about 1987 that the total number of protein kinase genes in a single organism would be will be rather high. You can see, starting with the first kinase, phosphorylase kinase in 1959, pKa in 1968, the numbers kept on increasing almost exponentially, particularly after the advent of cloning and sequencing. And it was on this basis that I made the prediction in 1987 that perhaps the mammalian genome might encode as many as 1,000 different protein kinases, and the protein kinase might be used in cellular regulatory circuits in a fashion analogous to transistors or chips in computers. And I think the subsequent 25 years or so have shown that, in fact, protein kinases and phosphatases do play key roles in cellular regulatory circuits. And I was going around and talking about this idea, and I was at one meeting when one of the speakers drew a sketch of me with kinases tumbling out of my beard here. The first kinome, that is the complete collection of protein kinase genes in an organism, was that for budding yeast, largely the work of Greg Plowman, who did all the bioinformatics. Once the complete sequence of the S. cerevisiae genome became available, and we deduced that there were about 115 protein kinase genes out of the 6,000 or so genes in this particular genome, and that allowed us to create the first kinome tree of these 113 protein kinases with major groups that Steve Hanks and I had proposed based on our sequence alignments earlier. All of this ultimately led to the human kinome, largely the work of Gerard Manning, who's now here, but was at Sugen at the time. So this is the human kinome. There are 478 protein kinases in the major part of the tree, and there are also a number of atypical protein kinases that are either not related or only weakly related in sequence to this major family. And here are the tyrosine kinases. They form a major branch of the tree, and there are 90 tyrosine kinase genes in the human genome. So since 2002, when that human kinome paper was published, there have been a number of revisions, and Gerard Manning now estimates that there are 538 protein kinase genes in the human kinome. So there's just been a small increase with some new remote and atypical kinases, but in fact, no new tyrosine kinases. And 3 kinases that we had listed earlier have now been disqualified for various reasons. So I think the kinome is important because it accounts for all protein phosphorylation events that occur in cells. Many of the 538 protein kinases are implicated in human disease. Gerard estimates at least 173, so over a third of all human protein kinases have been implicated either through mutation or over or under expression in human disease. And knowing the complete kinome allows one to carry out kinome-wide analysis, and there are now endless papers using si or shRNA knockdown of kinases to deduce their function in cellular processes. It also allows one to learn a lot about the evolution of protein kinases and protein phosphorylation in general, and Gerard has pioneered in this area and has really deduced the gains and losses of protein kinases, starting with the proto-eukaryotic cell. And finally, it's the largest family of enzymes and is of interest from this angle as well. So I thought I'd go back even further, though, and talk for just a little bit about why nature chose phosphate to make macromolecules, such as DNA, and use it to modify proteins in a reversible fashion. So phosphate's important, as was pointed out in Business Week in November last year. It's used for many, many things. It's in Miracle-Gro plant food, so the plant biologists are obviously happy about that. It's in Coca-Cola. That's what rots your teeth, as you probably know. It's used as fertilizer, et cetera, et cetera. But interestingly, in this long list of uses of phosphate, there was no mention of the fact that phosphate is essential for life. So, what is phosphate? Well, this is the periodic table. Many of you probably have not looked at this for many years, probably. Can you spot phosphate? Here it is. It's in group 15. This is the atomic structure of phosphate. You remember how this works? Protons and neutrons in the nucleus of the atom, and these are the electron shells here. Phosphorus has 5 electrons in its outer shell, so it can form 5 bonds by donating these electrons. So, for instance, it can combine with 4 oxygen atoms to form phosphate, and that's the structure of phosphate, also seen here. So there's a double bond to 1 oxygen and then single bonds to 3 hydroxyl groups. So as you're probably aware, phosphate's highly soluble in water, and it has a large, because of the oxygens, a large hydrated ionic shell. It's very abundant in nature, although it turns out that rock phosphate, which is what is mined for fertilizer, is running out in the world. So we're going to have to do something about that. Phosphate, as you all know, is critical for many cellular molecules, and phosphate esters and anhydrides are predominant in living organisms. So one of the keys to phosphate is that it has 3 ionizable hydroxyl groups, has 3 pKa's, so I think that's on the next piece here. So there's one that's basically always ionized, always a negative oxygen ion. One that has a pKa close to physiological pH, and that's important because it may or may not be charged in the cell. And one that's almost always a hydroxyl group. Has a very alkaline pKa. It's reactive at physiological temperatures. Obviously, that's important. It can form mono and diesters with alkyl and aryl hydroxyl groups. But it can also form acid anhydrides, such as found in ATP, the alpha, beta, and beta gamma bonds. And mixed anhydrides, as in PAPS, this is the sulfate donor for sulfation reactions, can also form phosphoramidates and phosphonates. This linkage here, the PC linkage, is non-hydrolyzable. It's found in some plant compounds. So in biology, phosphate obviously is important in ATP, which itself is a highly soluble molecule, 3 phosphate molecules in it, with free energies of hydrolysis of around 10 kilocalories, so this is an energy-rich compound. Interestingly, though, the phosphate esters to the amino acids, for instance, also have relatively high energy. So one of the keys to phosphate is that when it forms a diester, it can still have 1 ionized hydroxyl group. And this is important for retaining molecules like nucleic acids inside cells. So you can see, for instance, in a phosphate diester, such as in DNA, with these 2, say, in this case, bases linked to it, there's still a highly charged oxygen atom here with a very low pKa. So this retains then phosphorylated molecules, or molecules containing phosphate esters inside a lipid membrane, phospholipid membrane. So that's true for any molecule with a phosphate ester. And the energetics of phosphate anhydride and ester formation are favorable and occur under physiological conditions when catalyzed by protein enzymes. So obviously I wasn't the first person to think about this issue. And Frank Westheimer wrote this very perceptive article about 25 years ago now about why phosphate. But interestingly, even though phosphorylation, protein phosphorylation, had been known for about 25 years before this, he didn't mention protein phosphorylation in his review. So what about phosphate and protein phosphorylation? It's clearly one of the most common post-translational modifications. There are thousands of phosphosites occupied in any particular cell at any one time. And the phosphate esters of serine, threonine, and tyrosine have good chemical properties. They're very stable in aqueous solution at physiological pH. I think the key here is that phosphates on proteins form new chemical entities that are not mimicked by any natural amino acid. So aspartate and glutamate have negatively charged carboxylate groups as side chains, but their pKa's are different and their atomic radius are very different in terms of the number of coordinated water molecules from phosphate. And so that's probably why aspartate and glutamate often do not act as good phosphomimetic mutations, because they don't really have the same character. They have a single negative charge rather than phosphate, which usually has 1.5 charges, and they have this small ionic shell. In addition, phosphoamino acids form stronger salt bridges with arginine in particular, and hydrogen bonds. So that's very important in terms of their interactions with other proteins. So this allows then phosphoamino acids to be recognized by specific phosphobinding domains like SH2 domains, FHA domains. I should point out just in passing that although we mostly think about serine, threonine, and tyrosine phosphorylation, 9 of the 20 amino acids can be phosphorylated. And phosphorylation of histidine, I think, will emerge and is beginning to emerge as another important type of protein phosphorylation. So phosphorylation is a reversible reaction. It's readily reversed by hydrolysis, and that releases the free energy of the phosphate ester linkage. And so this particular reaction is not readily reversible. I just want to point out one other thing. So this is the reaction that creates a phosphoprotein. You have a protein, ATP, and an enzyme, a kinase, which generates a phosphoprotein and ADP. And this reaction is actually quite reversible. The equilibrium constant is between 2 and 50. And so you can actually drive this reaction backwards. If you add ADP and a kinase to a phosphoprotein, you can get specific dephosphorylation occurring. And that could be a useful tool, actually, to try and identify which sites are phosphorylated by which protein kinases. And then finally, phosphorylation is a highly versatile mode of reversible protein regulation. And we know examples where phosphate can act sterically or ionically to regulate function, can elicit allosteric conformational changes, as was in fact the first example of protein phosphorylation with glycogen phosphorylase, where the addition of a single phosphate greatly activates the enzyme through an allosteric change. And finally, and I think probably most importantly, phosphates can create a phosphodependent interaction through phosphate-specific binding domains that recognize phosphate in sequence-specific contexts. So phosphate has almost the ideal chemical properties for the formation of biological polymers and for the modification of proteins. And I would argue life as we know it could not have evolved without phosphate. Although I'll tell you something in a minute which might argue against that. So why phosphate and why not one of the closely related elements surrounding phosphate, like sulfate? Sulfate can form, sulfur can form sulfate with 4 oxygen atoms. However, sulfate only has 2 ionizable oxygens, both of which are very acidic. Meaning that sulfate monoesters always retain a single negative charge, but sulfate diesters have no negative charge. And sulfate diesters have an undesirable property of alkylating biological molecules. Silicate silicon on the other side, it's very abundant, obviously, but its lowest pKa is 9.5, and its esters are very unstable in solution. Now, arsenate's in the same group 15 here, arsenic rather. Arsenate has many of the same properties as phosphate, and many of you may have seen this paper in Science. It was published online on December the 2nd, but not in print until 6 months later. with 8 technical comments added to it. What these authors claimed is that they had identified a bacterium from Mono Lake in Northern California that could grow on arsenic instead of phosphorus. Obviously, that was somewhat heretical. So what they claim is that arsenate is incorporated into DNA, RNA, lipid, and protein, based largely on labeling of the radioactive arsenate, but also physical analysis. And they showed that these bugs will grow in medium containing no added phosphate, but 3 or 5 millimolar arsenate. However, their physical analysis showed that the DNA they isolated from these organisms still had a significant amount of phosphate in it, presumably coming from contaminants in the chemicals they used to make up the medium. And so this has been really the source of the debate is, are these bacteria really using arsenate instead of phosphate in their DNA, or is something else going on? There are good reasons to think that arsenate really wouldn't work very well as the basis for nucleic acids. It has the same sort of 3 pKa's, but the stability of arsenate esters and diesters is extremely low. So half-lives for an arsenate triester in water at pH 7 and room temperature is 0.02 seconds. So obviously that wouldn't be a good chemical basis for making a polynucleotide. They didn't actually demonstrate the existence of arsenate diesters in nucleic acids, and I think that was the key to most of the comments. They didn't show there was nucleotides containing arsenate instead of phosphate. I won't go through this much further. There are some other chemical differences. Bond lengths are different, and the partial negative charges on the oxygen atoms are different, and the radius of the arsenic oxygen bonds is somewhat different, which would distort the nucleic acids as well. So I think the bottom line is that it's unlikely that arsenate could have been used instead of phosphate, or is being used instead of phosphate as the basis for nucleic acids. But no doubt time will tell as people get these bacteria and begin to do further tests on them. Okay, so now I want to talk about some of our own work, and particularly about crosstalk between different types of post-translational modifications. Covalent modifications of proteins, like phosphorylation, clearly greatly expand the repertoire of the cell's proteome. As you know, there are only around 20,000 genes in mammalian genomes, and even with alternative splicing, this is not a very large number of protein entities. But if one adds in the number of possible modifications that can occur on a single protein, this number greatly expands. And as I have already told you, in the case of phosphorylation, many of these post-translational modifications are not only reversible, but are used to induce specific protein-protein interactions. So phosphorylation is recognized in a sequence-dependent manner by many different types of modular phosphobinding domain. The same is true for ubiquitination and SUMOylation. These are small proteins that are added to lysine residues. And also methylation, acetylation, and other types of modification can be recognized in this way. Hundreds of different post-translational modifications are known. We only work on a few of them, but there are hundreds of them, many of which we know very little about. I think it's still true that phosphorylation is the commonest post-translational modification, although with proteome-wide studies using mass spec, acetylation is certainly catching up pretty fast in terms of the number of sites that are modified. So there is a significant degree of crosstalk between different post-translational modifications on the same protein, and I'm going to talk a little bit about crosstalk between phosphorylation and ubiquitination and SUMOylation, and between ubiquitination and SUMOylation. So there are many ways in which phosphorylation and ubiquitination crosstalk, and this is from a review that I wrote a few years ago now. I won't really take you through this, but in many cases, phosphorylation of a protein can create what's called a phosphodegron. That is a recognition site for an enzyme, an E3 ligase, that can add polyubiquitin to a protein, which then triggers its degradation. Phosphorylation of the E3 ligase itself can also regulate its activity. And protein kinases and phosphatases themselves are subject both to activation by ubiquitination, but more commonly when they are activated through an upstream signal, they become targets for degradation, switching their activity off. Ubiquitination is linked to other types of post-translational modification. I've already told you now about how phosphorylation can drive ubiquitination. Here's an example. I'll tell you a little bit more about how SUMOylation of a protein can lead to ubiquitination. Glycosylation, so sugar additions, can also trigger ubiquitination. Hydroxylation of proteins, proline hydroxylation, is another target for inducible degradation. And very recently, this may be of interest to Bev Emerson if she's in the audience, ADP ribosylation has been shown to be a signal for regulated ubiquitination of proteins. So let me then talk about some of our own work, particularly the work of Huayu Sun on crosstalk between ubiquitination and SUMOylation. So these are two small protein molecules. I'll tell you a little bit about how they're added in just a second. One means of crosstalk is that the same lysine residue can be modified. So if you put a SUMO on a lysine residue, that prevents ubiquitin going on that same lysine residue and vice versa. And there are some examples of that. SUMOylation can target a protein for polyubiquitination, and I'll talk about RNF4. SUMOylation can regulate the activity of enzymes that are involved in adding ubiquitin or taking it off. I won't actually tell you about those. And SUMOylation itself, although I didn't mention this, can be triggered by phosphorylation. So ubiquitin is a small 76 amino acid protein. It's added to lysines or sometimes to other amino acids or the N-terminus of proteins through a 3-enzyme cascade. Ubiquitin is activated by the E1, an ATP forming an E1 ubiquitin conjugate. Ubiquitin is transferred to an E2. So there are 2 E1 enzymes in cells. There are probably 30 or so E2s. And then the E2 can either transfer its ubiquitin to a 3rd enzyme, an E3 ligase in the HECT family, and that ubiquitin is then transferred onto protein substrates. Or the E2 can interact with a ring domain protein which binds a substrate, and the E2 transfers the ubiquitin onto the substrate protein. So there are 2 main types of E3 ligase, the enzymatic ligases and the adapter ligases. And ubiquitin can be added either as a monoadduct or as a chain of protein of ubiquitins, which are these are the signals for degradation by the proteasome. But these chains can also be built not only through lysine 48 linkages, or here they are, lysine 48 linkages, but lysine 63 and several other lysines, and these have different functions in the cell. SUMO is related to ubiquitin distantly, but the fold is the same, and it's added also through a 3-enzyme cascade in very much the same manner, ending up with a SUMO on a lysine residue. And like polyubiquitin chains, SUMO-polySUMO chains can also be formed in cells. And SUMOylation has many functions in cells. I certainly won't take you through this, but one that's particularly interesting for what I'm going to tell you is double-strand break repair involves SUMOylation of repair proteins. But SUMOylation has a wide range of functions in cells, and probably many more that we have yet to learn about. So to talk specifically then about RNF4, it's a conserved family of ring domain E3 ligases with a C-terminal ring domain and 2 to 4 upstream motifs called SIMs, or sumo-interacting motifs. So these are short sequences that interact with sumo to form a non-covalent protein-protein interaction. And work from our group, particularly Huayu Sun and Nick Body at Scripps and several other groups, showed that these RNF4 family E3 ligases act to target SUMOylated proteins, probably polySUMOylated proteins, which they recognize through their SIM domains and then ubiquitinate through their ring domains. And work from Ron Hay and Hugh Detay's groups showed that RNF4 is required for the turnover of the PML nuclear body. It's a discrete nuclear structure. And RNF4 targets polySUMOylated PML for ubiquitination and degradation. Now, work from Huayu showed that genetic disruption of the RNF4 homologs in fission yeast, and this is also true in budding yeast, it turns out, causes genomic instability. And this fits with the idea that SUMOylation is integral to DNA repair. And work from Ron Hay and Steve Jackson has shown that siRNA knockdown of RNF4 in mammalian cells delays the resolution of DNA damage foci. So this is the structure of SUMO. It's, as I said, very similar to that of ubiquitin. And the SUMO interacting motif is not really not a domain in the normal sense. It's just a very short motif which contains a hydrophobic core and flanking acidic residues. And so in this brown color here, you can see a SIM interacting with the surface of SUMO, adding one extra beta strand to this beta sheet here. So Huayu and Joel Leveson, a former postdoc in the lab, showed that if one deletes both copies of the RNF4 orthologs, RFP1 and RFP2, in fission yeast, you get cells that look very unhappy. They're elongated, which means they're checkpoint arrested. They have cut nuclei where the DNA stain is not equal in two daughter cells. And most of these cells go on to die. So it's clearly required for genomic stability. And the two yeast orthologs are structurally very similar to the mammalian ortholog, RNF4. The C-terminus here is the ring domain. They're obviously closely related. The ring domains have these characteristic cysteines and a histidine that coordinate two zinc ions. And they also have this highly conserved C-terminus with 3 hydrophobic residues and a central aromatic. And 2 to 4 of these sumo interacting motifs at the N-terminus. The yeast proteins have 2 and the mammalian proteins have 4 of these. So this is not only conserved in sequence but in function because it's possible to complement the double deletion strain by expressing mammalian RNF4 in these cells. So you can see the cells without lacking both of these RFP genes doesn't really grow in this test here. Whereas if one expresses the mammalian RNF4, cells grow fine, just as they did if you put back RFP2. And if one mutates either the central SIM domains or the C-terminal ring domain, you lose complementation. So that says this is a conserved function that requires both the SIM domains and the ring domain. And this just demonstrates that RNF4 itself has this sumo-targeted ubiquitin ligase or stubble activity. So here we've used an artificial substrate which contains 2 tandem sumo residues and a fused GST. And you can see that the wild-type RNF4 in this ubiquitination assay where we've added ubiquitin E1 in E2 forms a ladder of ubiquitin adducts, whereas the delta sim RNF4 doesn't do that. So we were curious about exactly how RNF4 ring domains acts as an E3 ligase. And several prior structures of ring ligases show that these act either as heterodimers or in some cases as homodimers. And in the heterodimers, only one of the two ring domains is active. So for instance, BRCA1 and BARD1, as work we did together with Inda Verma showed, the BRCA1 ring domain is active and the BARD1 ring domain is not, but helps the BRCA1 ring domain. The same is true for MDMX and MDM2, one of Jeff Wall's favorite proteins. So together with Catherine Day and Shu Liu at the University of Otago in New Zealand, we solved the structure of the RNF4 ring domain. And like CIAP2, it's a symmetrical dimer, and that highly conserved C-terminus lies in the middle between the two ring domains, and so plays a key role in this interface. And by mutating residues that are involved in dimerization, we were able to show both biochemically that they cause the RNF4 ring dimer to collapse to a monomer, and that these mutations, such as this mutation here in the tyrosine in the C-terminal tail, abolishes E3 ubiquitin ligase activity in vitro. Huayu was also able to test the consequences of these non-dimerizing mutants in the S-POMBI assay, and particularly under conditions of genomic stress, where we've added hydroxyurea here, you can see that the wild-type RNF4 supports growth, whereas this Y193A non-dimerizing mutant fails to support growth. In addition, analysis of these same mutants showed that mutants that fail to dimerize, like this Y193A, also lack this in vitro stubble activity on this SUMO GST substrate. Let's skip that. So what we conclude then is that RNF4, like CIAP2 and many other ring domain ligases, acts as a homodimer. And in mammals, this is a true homodimer, whereas what I didn't tell you was that in the yeast, the RFPs interact with another ring domain protein, SLX8, to form a heterodimer, which appears to serve the same function. So we know that the ring is a dimer, and this is critical for function. It's possible that there are additional dimerization interactions in the full-length protein for which we do not have a structure. And it's certainly possible that polysumoylated substrates may facilitate the formation of an active dimer by binding to the sim domains on both molecules. We're still unclear exactly how or why a dimer is required and how it activates the E2 ubiquitin thioester linkage, which is the key to transferring ubiquitin onto a substrate protein. We still don't know how the polysumoylated target protein is selected by RNF4, how it's really bound to the sim domain. In other words, do multiple SUMOs have to interact with different SIMs for this to occur? And is it presented in cis or in trans within the dimer? And we're still looking for the key polysumoylated protein targets that are involved in genomic stability. There are certainly some ideas out there now, but in the yeast we really don't have a very good handle on what these proteins might be. So Wai-Yu was interested whether there might be additional stubble-like proteins in, particularly in mammals, and so he developed a way of bioinformatically screening for proteins that contain multiple RNF4-like SIMs. So he basically built a motif search that could look for proteins with at least 2 motifs of this type. And we think it's important to look for proteins with multiple SIMs because these can form domains that interact either with multi-SUMOylated or poly-SUMOylated proteins or with SUMOylated protein complexes. So he wrote this program, SimSearch, in Python script, screened the human RefSeq protein sequence set of 38,000 sequences and came up with 89 proteins that have 2 matches. Gratifyingly, one of these was RNF4, as you would have expected, but there was a second ring finger protein, RNF111, also known as Arcadia, which has a tandem array of 3 SIM motifs and a C-terminal ring domain. So it's a much larger protein than RNF4, but potentially could have a similar function. So he showed by analysis of SIM domain mutants that SIMs 1 and 3 are important for binding SUMO in vitro using GST fusion SIM domain, wild type and mutants, and flag tagged SUMO. Particularly, I think SIM3 is the most important of the 3 of them. He showed that the SIM domain of Arcadia, as I'll call it, was able to interact with poly-SUMO chains in vitro, and that Arcadia has stubble activity, although it's relatively weak, in vitro, again using this artificial substrate. So you can see the ladder here of ubiquitinated SUMO GST that is lost with the SIM mutant or the ring domain mutant. Now, Arcadia is an interesting protein. It's already been quite well studied. It's an essential gene in the mouse, and the knockout is an embryonic lethal due to a failure to form the node. And work from Episcopu and other groups has shown that Arcadia is critical for enhancing TGF-beta signaling during development. And there is evidence that Arcadia promotes the degradation of 2 negative transcriptional regulators of TGF-beta response genes. So Wai-Yu tested whether the SIM domains, SIM motifs, are important in Arcadia stimulation of TGF-beta signaling by reconstituting Arcadia knockout MEFs with either wild-type Arcadia or SIM mutant or ring domain mutant. And then stimulating these cells with TGF-beta and looking at the expression of PAI1, which is a target gene for TGF-beta using qPCR. So in the wild-type MEFs, you can see a nice TGF-beta stimulation of PAI expression. In the knockout MEFs, you see a reduction. There is still some induction, so TGF-beta response is not totally dependent on Arcadia. If you express wild-type Arcadia in the knockout cells, you see actually now a very strong TGF-beta response, greater than you see with the wild-type cells. And this is reduced somewhat by the SIM mutation and abolished with the ring domain mutation. So I think what we conclude is then that the SIM domains can play some role in promoting TGF-beta gene expression. And several proteins in the pathway are SUMOylated, and Huayu is now investigating which, if any, of these might actually be targeted for ubiquitination by Arcadia. I think I'm going to skip this because I want to get on to one last thing. What those two slides showed was that potentially Arcadia, like RNF4, may have a role in reactivating methylated promoters, and so deeply repressed promoters may be targeted by Arcadia, and this could be an explanation for why some TGF-beta target genes are sensitive to the expression of Arcadia. So what we conclude then is that stubble activity is not unique to RNF4 family. There's at least one other E3 ligase that potentially has stubble activity, and Huayu has some evidence for a third one. It is possible to identify novel SUMO binding proteins computationally, and I think what we're learning is that this SUMO ubiquitin crosstalk is becoming increasingly complex. So in the last 5 minutes, I know I'm going to run just a little bit over here. I want to just turn back to the kinases and talk about some recent work in which we've tried to identify novel protein kinases that might play a critical role in cancer. Traditionally, as I pointed out at the beginning, many of the protein kinases involved in cancer were found through the study of retroviral oncogenes, which had captured these kinase genes and activated them. In addition, oncogenic protein kinases have been identified as the fusion partners for chromosomal translocations, such as BCR-ABL. But recently, with the advent of whole genome sequencing, it has become clear that there may be many more protein kinases that are mutated in cancer, and some of the early studies from the Sanger group showed that BRAF, a member of the RAF family of serine-threonine kinases, is commonly mutated in melanoma and in colon cancer. And it turns out that these mutations are activating mutations, and there are now 2 drugs in late-stage clinical trials targeting the mutant form of BRAF that appear to be quite successful in delaying the progression of melanoma. But as I said, there are many protein kinases that have been now reported through these sequencing efforts to carry mutations, albeit at relatively low frequencies. And the question is, which of these mutant kinases actually play a causal role in cancer? And would they be good kinase inhibitor drug targets if they do? So this is a table from one of the Sanger Center sequencing papers. I don't want to take you through this. They sequenced, they found more than 1,000 somatic mutations in 518 protein kinase genes that were analyzed in 210 different cancers that they analyzed. And there were putative driving mutations in 120 kinases. Now, by putative driver, this is all based on a statistical analysis based on synonymous versus non-synonymous mutation ratios. At the top of the list is BRAF, which I've already told you about, and ATM, which is a tumor suppressor protein and these mutations inactivate ATM. So John Brogdon, when he was a postdoc in the lab, went through several of these cancer kinome sequence databases and resorted or reanalyzed the mutations for their probability, not only of being a driver mutation, but of being involved in cancer using a couple of different web-based bioinformatic applications. And this was the rank order he came up with of kinases, DAPK3, serine-threonine kinases, FA3 as a receptor tyrosine kinase, etc. And he chose to begin to study 3 of these, all serine-threonine kinases about which little is known, to characterize the cancer mutations in the hope that we'd learn not only something about their role in cancer, but also something about the normal function of these protein kinases. So I'll just tell you now about work on DAPK3, death-associated protein kinase 3. It's a member of a family of 3 protein kinases in the larger CAM kinase family. They all have very closely related N-terminal catalytic domains. Their C-termini, as you can see, are quite different. Very little is known about DAPK3, but it can heterodimerize with the other 2 members of the family through its catalytic domain, as I'll show you. A few substrates have been reported, and we'll be using myosin light chain 2 as a surrogate substrate for our assays. And there are rare mutations that were identified by sequencing in breast, lung, and ovarian cancer. Here are 3 such mutations, all lying in the catalytic domain. We know where they lie on the catalytic domain because the structure was reported by Lawrence Pearl's group. These 3 residues are conserved in all 3 of the DAP kinases. And this actually shows you how the catalytic domain forms a strand exchanged dimer in which the activation loop from one catalytic domain inserts into the other catalytic domain, and this is required for the phosphorylation of this key activating threonine residue. So John developed a strategy then to analyze the consequences of these cancer mutations. First, he analyzed the effect of the 3 mutations on in vitro kinase activity of flag-tagged proteins isolated from transfected 293 cells using GST MLC2, myosin light chain 2, as an in vitro substrate compared to either wild type or an activated C-terminally truncated mutant, and the T180A activation loop mutant, which is dead. You can see all 3 mutants have lower or no activity in vitro compared to the wild type and delta 273 mutant. In vivo, when he overexpressed the 3 mutant kinases and the wild type and the activated mutant increase the level of phospho-MLC2 in the cell, whereas the 3 mutants, with the possible exception of this T112M, do not. So we conclude then, based on these in vitro and in vivo studies, that these mutants have reduced catalytic activity. He then tested whether they affect cellular functions. The wild-type protein and the delta 273 can both delay the cell cycle when overexpressed by increasing the G1S ratio. The 3 cancer mutants do not. The wild-type and the delta 273 can increase cellular apoptosis when overexpressed at low serum levels. The 3 cancer mutants do not, so they appear to be non-functional by these assays. And then, because I didn't tell you this, we tested whether they could act as dominant negatives, since these cells, the cells that were, and tumors that were sequenced, all carry these mutations as heterozygous mutations. And so if they are to have a function in the cell or an effect in the cell, one would expect them to act in a dominant negative fashion. And what he found was, by expressing the wild-type protein, either alone or together with an equal amount of each of the mutants, that the 3 mutant proteins, if you just look at these 3 lanes here for phospho-MLC2, were able to suppress the ability of the wild-type to enhance phospho-MLC2. And we think that this is because the mutant proteins can heterodimerize with the wild-type protein, both through this catalytic domain strand exchange, as well as through a potential leucine zipper at the C-terminus. And consistent with this, we were able to show, and I won't take you through these data, that the wild-type protein was able to co-precipitate the 3 cancer mutant proteins. So they can form dimers in vivo. I think I'll skip that, which showed that the mutants can override the ability of the wild-type protein to delay DNA synthesis and cause apoptosis in cells. Finally, then, we set out to test whether the cancer DAPK3 mutants could play a causal role in tumorigenesis, where there is a single cell line carrying a mutant DAPK3 together with a wild-type allele. We have tried so far unsuccessfully to selectively knock down the mutant allele through siRNA. You only have a single mutated residue to target, so it's not trivial. And currently we're considering whether to carry out knockout by homologous recombination. But we did do one experiment which is suggestive, and that is that if one expresses wild-type DAPK3 in these cells with the endogenous DAPK3 mutation, then their growth as spheroids in culture is significantly reduced, suggesting that this may be overriding the effect of the mutant. So I'll just conclude then. The mutations we've analyzed in DAPK3 that were identified through the cancer kinome sequencing efforts cause a loss of function. One of these we might have predicted anyway because it's in one of those motifs that I showed you at the beginning to be essential for catalytic activity. These mutations have functional consequences and appear to override the ability of the wild-type DAPK3 to act as a tumor suppressor. Consistent with the role of DAPK3 as a tumor suppressor, ZIP kinase, another name for DAPK3, was shown to be underexpressed in about 2/3 of aggressive gastric cancers by a Japanese group. So what we still really need to show, though, is that the DAPK3 mutant alleles do play a key role in tumorigenesis. And if they do, what particular signaling pathways downstream of the wild-type DAPK3 are they interfering with? And we think potentially autophagy is one of these functions. So to conclude, not only are the mutations in DAPK3 inactivating, we have also found that a series of mutations in the protein kinase C family identified through sequencing efforts are also loss-of-function mutations. And it's beginning to appear from our work and that of others that cancer-associated kinase mutations may commonly be loss-of-function. Prior work on CFS has reported this. A recent paper on MAP2K4 and unpublished data on FA3 and ARAF suggests that these cancer mutations are all inactivating. And in consequence, these kinases, although they're playing a role in cancer, will not make good drug targets. So with that, sorry I ran on too long. I'd just like to thank the people who did the work. The work on RNF4 was largely carried out by Huayu Sun, and the work on the cancer-associated kinase is largely by John Brognard. Thank you very much.
Okay, have we got any questions? Maybe I can start, Tony. So with the DAPK mutants then, the fact that you don't get loss of heterozygosity, do you think that they actually not only have a loss of function, but a gain of function as well?
Yeah, it's possible that there is a gain of function. And that's something we really haven't been able to get a handle on, but I would agree that the heterodimer with the wild type may be doing something in addition to just loss of function.
Right.
And the real problem is we know so little about the DAPK3 downstream signaling pathways.
Right.
So there's recent evidence it may be connected to autophagy, and that's something that's certainly be worth looking into.
And is the dimer more stable just of the mutant and the...
Right, so we don't really know. I mean, we assume that the levels of the wild type and the mutant are the same in the cell, but that may not be true. And we've not really We don't really know what fraction of the wild type is in a heterodimer. On theoretical grounds, if the proteins that are the wild type and the mutant are expressed equally, we would expect 75% of the wild type to be present as part of a heterodimer, but we really don't know this. That's something, if we had allele-specific antibodies, we might be able to test that out. When we can do things with overexpression, it looks like they basically form a stable heterodimer, but in the real cell, I don't know. Oh.
Yep.
Would it be fair to ask something about the P53 gene? In other words, 4 or 5 years ago, that was supposed to be the watchdog for most all cancers. Can you comment on that? And also, I'm curious as to why they never found a marker for ovarian cancer before it reaches the peritoneum. And also, why the failure for a vaccine for melanoma, which Don Morton has worked on for years. I just wondered if you have anything to fill in there with more details as to why we're having such a struggle with these things. I mean, maybe this is not a fair question, but...
I don't work on any of those things. I think P53 remains an extremely important gene in cancer, but cancer cells have many mutations, and so the loss of P53 clearly engenders genomic instability, which allows other mutations to accumulate, but that by itself doesn't drive cellular proliferation. You need these other mutations, maybe dependent on the loss of P53, to drive cellular proliferation. The inability of a vaccine against melanoma to be very successful, I think, it just illustrates the fact that tumors are very successful at escaping the immune response. Even though melanomas do express apparently unique surface antigens, if you put enough selective pressure on them, they will lose that surface protein because it probably isn't relevant to the function or to the properties of the tumor cell. As regards to the ovarian cancer markers, I agree, it's surprising one hasn't been able to find anything. I think the hope is with continued molecular analysis, one will find something that is unique to ovarian cancers that maybe can be detected in circulation as methods get more sensitive for doing mass spec analysis and things like that.
Terry.
So you gave a long list of potential mutations from the human cancer genome, and you focused on one. Are there any other promising ones on that list that are emerging? Maybe not from your lab, but from other labs.
So are there? Well, one of the. I mean, I already talked about what we had worked on, but I would say that ALK, which is a receptor tyrosine kinase, emerged from this type of genomic analysis. It wasn't actually sequencing per se, but it was similar sort of kinome-wide analysis. And mutations were identified in about 5% of non-small cell lung cancers. Several pharmaceutical companies have now developed ALK-specific inhibitors, which are looking quite promising in that subset of non-small cell lung cancer. So it was, I think there will be other success stories out there. The issue is going to be, if the frequency is very low, will there be significant interest in developing small molecule inhibitors or protein inhibitors for that matter? And drug companies are generally loath to develop anything unless there's going to be 1 million patients. In the case of Gleevec, it was interesting because they put it on the shelf. They were not interested in developing Gleevec, but ultimately, I think Brian Drucker persuaded their CEO that it would be good publicity to do this, even if it wasn't going to be profitable. But what they hadn't realized or thought about was the fact that if it was successful, they would have only while they might have only 10,000 patients in the first year, they would have 20,000 in the second year, 30,000 and so on, because patients would stay on the drug for their life. And so in the end, it's turned out to be a multi-billion-dollar drug, even though they had not predicted this. So I think the hope is that in other cases, this will also turn out to be the case, that the drugs will be used for sustained periods, and therefore companies be willing to develop them. Yes, Inder.
Disappointing that so many kinases turns out to be loss of function than a gain of function. But there is a lot of homology among these kinases. is it sensible to start thinking that if, let's say, one of them, which is a loss of function, then one should look for another one, very similar in its activity, and activate its activity, which seems to be a lot easier to do than to deal with something which is a loss of function. Is that something people are thinking about, or is that something worth thinking about, using, activators, acetylases, and so on?
Right, so Inder's right in the sense that all of these things lie in pathways, and if one kinase shows a loss of function, maybe one can find something downstream of it that one can activate instead. And I think that is going to be a good approach. If we understood the networks better, I think we could think about doing that. I mean, so it turns out that DAPK3 is closely related to one of the other kinases we have begun to study, SGK085. And we don't know whether maybe those mutations are activating in that in that setting. So I agree. I think, just because the kinase itself is not a target, but something in that pathway might well be a target for either an activator or even an inhibitor, I guess. We need to understand the logic of the circuits perhaps better to do that rationally. We've already had a number of surprises, like with rapamycin, which inhibits the mTORC1 kinase, and it's very effective at doing that, but it turns out that inhibitor really leads to the activation of a downstream kinase. And so you get unexpected consequences sometimes of inhibiting a kinase, which actually really fits with your idea.
I was just to expand, thinking more about synthetic lethality, like with the KRAS, and so I think I'm just trying to think, is that really a big effort at the moment, or is that something just occasionally when people have a good screening process, they do it?
Well, I think there's a lot of interest and probably activity in doing synthetic lethal screens with different kinases. And it's a big matrix if you have to do a synthetic lethal screen with 500 and something kinases against 500 kinases, but I think people are beginning to do that. Ed Haller did a very interesting siRNA screen in a series of tumor lines where he found even in tumors, lines derived from the same tumor, that different kinases were essential genes. And so I think there will be a lot of interest in that type of approach.
Tony, histidine phosphorylation is important for a lot of lower organisms, but have we outgrown it, or is there a reason why we haven't found it in eukaryotic organisms?
Phosphorylation?
Of histidine residue, sure.
Ah, yes, well, I sort of skipped over that. So in bacteria, histidine phosphorylation is used as a means of signal relay in two-component systems, which generally are involved in sensing external stimuli. And these are very simple systems in which one protein autophosphorylates on a histidine when it receives a signal and transfers that phosphate onto an aspartate residue, which is the effector molecule, often a transcription factor. So that type of histidine kinase is found in a few simpler eukaryotes. There are some in plants, there are 1 in yeast, 4 in dictyostelium, but no metazoan has that type of kinase. So instead, in metazoans, there appears to be a different type of histidine kinase that actually transfers gamma phosphate of ATP onto a histidine residue in a target protein. And there's some evidence that might be a reasonably common event. The problem is that histidine phosphorylation is relatively unstable chemically, and so it's hard to study. It falls apart at pH 7 or below, and so you have to work at more alkaline pHs to study it. So I think it's clear there won't be any of the prokaryotic style histidine kinases, but there may well be a different type of histidine kinase that's important. And Ed Skolnick's group has already shown that histidine phosphorylation of a potassium channel is a way of regulating channel opening.
Okay, well, with that food for thought, I think it's time for lunch. Let's thanks to Tony.